Assalamualaikum warahmatullahi wabarakatuh. My name is Nurainna Fatiha binti Izzuwan and I'm a fourth-year student of Bioinformatics. My supervisor is Dr. Zuraini Alisha. Did you know that more Malaysian children are being diagnosed with autism spectrum disorder each year? According to the chairman of National Autism Society of Malaysia, Failing S.Y. Muhammad Faizul, by analysing the national gross birth rate per year, there are around 8,000 to 9,000 born yearly that may have autism in Malaysia. However, there is no official registry for the number of individuals diagnosed with autism in Malaysia. Meanwhile, WHO estimates that 1 in 160 children has ASD and its prevalence appears to be increasing globally. You might be wondering what are these facts has to do with my presentation. Hence, I would like to introduce my research title, which is SVM and MLP, a comparative analysis on autism gene expression data classification based on machine learning methods. Here is the outline for my presentation, where it is divided into 6 parts, which are introduction, literature review, research methodology, research design and implementation, result analysis and discussion, and lastly will be conclusion. For the first part, which is introduction, I will give a brief explanation about the domain in this study, which is ASD, gene expression data, and I will also state the aim and objectives. ASD is a lifelong neurodevelopmental disorder that could affect human communication and behavior, as it is related to social interaction, speech, and communication. This disorder makes it hard for them to relate to the outside world, and it is also hard for them to communicate with others. Aggressive and/or self-injurious behavior may be present in some cases. As for gene expression data, it is a set of data that holds significant information for the expression values of genes that are computed from the microarray technologies. The dataset is extracted from the gene expression process, where DNA is converted into a functional gene product. A lot of research has been conducted using various machine learning algorithms to study ASD. Gene expression in ASD is used not only in an attempt to separate ASD into different subtypes, it is also used to select the most attributed genes. However, the uninformative gene is also included in the dataset due to the high dimensionality of gene expression data. Regarding this problem, this research is conducted to classify the genes into autism and non-autism from the gene expression data using machine learning methods. There are 3 main objectives for this research, which are: first, to identify the classification method that can be implemented on autism gene expression data. Second, to propose FS1 support vector machine-based filter feature selection algorithm to improve the accuracy of classifier in ASD. Lastly, to evaluate the classification method implemented on autism gene expression data using accuracy performance. Classification plays an important role in organizing data. In this study, classification technique works by predicting the target class for each group of data, which are autism and non-autism. As for machine learning, it is a method of data analysis based on the idea that systems can learn from data, identify patterns, and make decisions. There are 4 machine learning methods used in this study, such as multilayer perceptron, key nearest neighbor, naive Bayes, and support vector machine. The first method is multilayer perceptron. It is known as feed-forward neural network that use backpropagation for classification. It also has more stacked layer in which every layer consists of small pieces called neurons. The structure of MLP consists of 3 main layers, which are input layer, hidden layer, and output layer. Those neurons get input data X in the input layer and then share that data to the next layer. In the next layer, which is a hidden layer, each neuron will receive a weighted sum of the neuron output the previous layer. In order to control the input, the activation function is implemented within each neuron. Next, key nearest neighbor, in which the basic concept is to predict the target label by finding the neighbor class closest to them. The implementation of KNN is based on 2 operators, which are the value of k that refers to the number of neighbors to consider and the distance measure used. The significant part in using K&N method is choosing the right number of neighbors. It is essential in order to maintain the ability of the algorithms to make a classification accurately. The third method is naive Bayes. Naive Bayes is a supervised machine learning algorithms that use Bayes' theorem, which assumes that features are statistically independent. The classifier is assumed to have no relation to other classes with other features in the class. Naive Bayes has the advantage of being probabilistic models, resilient to missing values and real data noise. Lastly, it is support vector machine, which classifies the data points by finding n-dimensional hyperplane, where n denotes the number of available features. A hyperplane is a decision boundary used to separate space in the input variable. SVM can exist in two categories such as nonlinear SVM and linear SVM. Nonlinear SVM is used for dataset that cannot be categorized using a straight line width, while linear SVM is used for data that can be separated linearly. SVM is widely used in classification of ASD due to its ability to provide a decent accuracy level in classification for high-dimensional data. The third part in this presentation is research methodology, which consists of research workflow and performance measurement. There are three phases available in the research workflow. Phase 1 is about research planning and data preparation. During this phase, various resources such as journals, conference paper, articles are collected. The aim, purpose of study, objectives, and scopes are also provided and continued with the planning of research flow. Other than that, the dataset are also prepared during phase 1. As for phase 2, it is divided into two parts, which are feature selection and classification. In phase 2.1, the algorithm for feature selection are chosen according to the previous studies that has been conducted. The feature selection method is then conducted and a number of features are selected to proceed with the classification. During phase 2.2, the four algorithms are chosen and implemented for classification. The parameter for each classifier is modified in order to get the best result. Lastly, phase 3 is about prediction assessment, in which the result for each classification method will be evaluated, analyzed, compared, and discussed. As mentioned previously, the performance of each classification method is measured through accuracy, precision, and recall. A true condition table can be referred to evaluate the performance. Accuracy can be defined as the percentage of correctly classified instances. As for the precision, it is also known as positive predictive value, where it simply shows the ratio of correct positive prediction out of all positive predictions. Lastly, recall is the fraction of the total amount of relevant instances. In this part, a research design and implementation will be explained in detail through data preparation, data characteristics, model parameter, and feature selection. Data preparation is the method of cleaning and transformation of raw data before proceed to processing and analysis. In this study, the data is retrieved from GEO repository with an accession number GSE25507. The dataset is prepared by an experiment profiling by array, in which microarray from gene expression were made on purified RNA from peripheral blood lymphocytes in children. Next, the dataset also undergo data reduction process, where the data is reduced from 54,630 number of features to 250 features using GEO2R. GEO2R allows users to make a comparison of two or more groups of samples in order to identify genes that are expressed differently. The comparisons are done using GeoQuery and Lima-R package from the Blood Bioconductor project, which is based on the R programming language. As for the normalization, min-max normalization is performed to overcome the problem of having a wide range of values in the dataset. Hence, min-max normalization is conducted to ensure that the dataset has a similar range so that each feature will be equally important. It works by transforming the minimum value of each feature into a zero and the maximum value is transformed into 1. This is the data characteristics after performing data reduction. The dataset accession number is GSE25507 and it is divided into two classes, which are autism and control. The dataset has 146 number of samples and 250 number of genes. The number of autism is 82 and 64 is for control. Machine learning models are parameterized so that the model can adjust their behavior based on the given problem. For MLP, the parameters are the number of hidden layers, which are set into 2. According to the research done by Copexco, one hidden layer is enough to get an accurate approximation of the model function. However, overfitting was shown to be more common in one hidden layer structure. Hence, two hidden layers are used in this study, which could get a better result. The activation function used for the hidden layer is rectified linear unit function, and the solver used is stochastic gradient descent. As for the number of maximum iterations, the default value is chosen, which is 200. This value also determines the number of epochs in which the number of times each data point will be used. In KNN, the model parameter is the number of neighbors, which is k. Generally, the number of neighbors chosen is usually an odd number, and the usual practice in choosing the value of k is using the formula k is equal to square root of n, where n is the number of samples in the training set. Hence, the number of neighbors chosen for this research is 9. Lastly, the parameters implemented for SVM are three different types of kernels. The first kernel is Gaussian radial basis function, which is the most used type of kernel, and it is useful in making a proper separation when there is no prior knowledge of the data. Next, linear kernel, which is the most basic type of kernel. It is more preferably used when the data is linearly separable. Lastly, polynomial kernel, where the degree of polynomial is set to 5. The result for the different kernel of SVM will be shown and discussed in the next slide. Feature selection is the process of selecting the characteristics that contribute the most to the prediction variable. In this study, feature selection is used to improve the result of SVM classifier since the classifier has obtained the best result among other classifier used. Lasso and Relief F are chosen as the feature selection method. Relief is an algorithm that uses statistical method that weighs each feature according to its relevance to the class. Relief F is an extension to Relief that introduces deterministic neighbor selection approach. It works by calculating a feature score for each feature, which can be applied to rank and select top scoring feature for feature selection. The number of features selected are 100 for this method. As for Lasso, it stands for Least Absolute Shrinkage and Selection Operator, which is a regression analysis method that is used to enhance the prediction accuracy. This model uses shrinkage, in which the data values are shrunk towards a central point as the mean. During the feature selection, the variables that still have a non-zero coefficient after the shrinking process are selected. Hence, there are 48 number of features selected in this method. The result of each feature selection method will be shown and discussed in the next part of this presentation. In this section, there are three different results that will be discussed, which are the classification result among 4 different classifiers, performance of SVM with different kernel, and lastly is performance of SVM with Lasso and Relief F. In this slide, it shows the classification result of SVM, Naive Bayes, KNN, and MLP for both 250 and full data. The result is compared with full data in order to get a different perspective for the classification result. Based on the table, it can be seen that SVM obtained the highest result in accuracy, which is 86%, compared with MLP, Naive Bayes, and KNN, which obtained 80%, 81%, and 83% respectively. However, this result contradict when using a full dataset where the highest accuracy is achieved by MLP with an accuracy of 100%. Both SVM and NIF Bayes obtained the same value of accuracy, which is 76%, while KNN achieved the lowest accuracy with a value of 63%. As for precision in 250 data, it can be seen that SVM also has the highest value, which is 89%, followed by NIF Bayes with a precision value of 85%, along with MLP and KNN, which obtained 81% and 84% respectively. The lowest value of recall is achieved by NIF Bayes with a value of 83%, while other three classification methods achieve 89% and 86%. In contrast, KNN achieved the lowest result for recall when the full dataset is used, which is 57%. This algorithm also obtained the lowest value for precision, which is 74%, while both SVM and NIF Bayes obtained 80% for both precision and recall. Whereas, MLP achieved 100% in both precision and recall, which make it the best classifier when the full dataset is used. MLP can be assumed to have a potential to become a great algorithm in classifying autism genes, as this algorithm has the second highest value of recall, which indicates that it has a great ability to correctly identify genes with ASD 250 dataset. However, this study focused on the use of 250 data, which is the dataset that has undergo data reduction process. Hence, we will emphasize the result obtained from 250 data, where SVM classifier has shown the best performance in terms of accuracy, precision, and recall. In this part, the dataset is run using three different kernels of SVM in order to evaluate the performance of this classifier. From the result recorded, the best kernel performance is polynomial kernel, which scored the highest value of accuracy, which is 88%. This significantly better result delivered compared to the other kernels might be due to the use of higher degree polynomial kernels, which are set to 9, resulting in a more flexible decision boundary. As for linear kernel, it has been chosen to run both 250 data and full data due to the fact that the number of features is larger than the number of observations. Lastly, the RBF kernel produced the lowest result of accuracy with a value of 85%. Feature selection is conducted to improve the result of SVM classifier for classifying autism gene expression data. According to the result recorded, there is a slight improvement for the performance accuracy by implementing Relief F, in which the result of SVM increased from 86% to 88%. However, there is a deterioration in the performance of SVM after implementing Lasso, where the result decreased from 86% to 83%. An increased performance of accuracy after implementing Relief F as feature selection might be due to its ability in using feature-weighted kernel function to reduce the weak correlation feature impact on classification result. Therefore, the performance of the classifier can be improved after implementing this method. In the last section of this presentation, there are three main parts that will be discussed, which are achievement of project study, limitation of study, and suggestion for future works. In general, all three objectives of the study have been achieved successfully. For the first objective, the methods used in this research are MLP, KNN, NIF Bayes, and SVM, where they are chosen based on several research studies that have been conducted by other researchers. To answer the second objective, two feature selection methods are implemented on SVM classifier and compared, where Relief F managed to improve the accuracy of the SVM classifier in ASD. As for the last objective, it has been achieved where the performance of each method has been successfully obtained and evaluated based on accuracy, precision, and recall. However, there are some limitations that have been encountered during this study. The classification methods used in this study are more focused on the identification of gene expression data in ASD. The results achieved for each method may be different when using other types of datasets such as personal characteristics data, structural MRI, or functional MRI data. There are some parts that can be improved for further research in the future. Therefore, some of the future works are provided that could be done for the enhancement of the study, such as conduct this study by using other types of dataset that is related with ASD, such as personal characteristics data, raw video data, structural MRI, or functional MRI data. Next, conduct classification method by focusing more on deep learning methods such as DNN, CNN, RNN, and GRU. Lastly, explore and implement other parameters for SVM classifiers such as gamma and C. All in all, this study can be used as a guideline in choosing the machine learning method for gene expression data classification in ASD from the numerous methods available. Given that the most performed classifier is SVM, the implementation of feature selection with the objective to improve the performance of SVM classifier has proven to be successful, where there was an improvement for the accuracy performance. Other than that, the accuracy result for MLP classifier also has shown a significant performance for the full-size data. Hence, this study can provide a valuable insight on which method to be used in classifying ASD genes into autism and non-autism. I think that's all from me. Thank you.